To finish up our study on carbohydrates, we now consider the topic of polysaccharides. So there are three important polysaccharides that we need to consider, and all of them are considered glucan, which means that they are made of glucoses joined together by glycosidic linkages to form long polymers. So the three polysaccharides that we're going to consider are starch, which, as we will see, is a mixture of amylose and amylopectin, glycogen, and cellulose. Okay, so starting with starch, starch is the principal food reserve of plants. Okay, so plants take their glucoses, and if they don't need them all, they store them as starch. And starch is made of 10 to 20% amylose and 80 to 90% amylopectin. So let's first look at what amylose is. So amylose is an unbranched polymer of D-glucose connected in alpha-1,4 glycosidic linkages. Okay, so what do I mean by this? Well, 1,4 means, in the terminology that I used in the previous lesson, head to tail. Okay, so the anomeric carbon of one glucose is hooked up to the carbon 4, the tail carbon of the other one, and the stereochemistry of the glycoside bond at the anomeric carbon, of course, is alpha. So there's two views of it here. We've got the Haworth view on the right, which makes it easy to see that from the perspective of the left-hand glucose, the acetal oxygen is pointing down, and of course the CH2OH coming off of C5 is pointing up, that alpha relationship. And then you could also see it in the cyclohexane chair conformations shown below. So it turns out that when you have this type of structure with all of these alpha linkages, the molecule, the macromolecule, is actually not linear. So it starts to develop a twist to it. So the macromolecular structure is that these alpha glycoside linkages induce a helical macrostructure that actually leads to a compact shape. So it forms a helix and twists around among itself. Amylopectin, on the other hand, is a branched polysaccharide. So structurally, it's very similar to amylose in that we have the alpha-1,4 glycoside bonds linking together the glucose units in the chain, but then the branching occurs between C6 of one glucose unit and C1 of another. So this is called an alpha-1,6 branch point because obviously with respect to one glucose, it's the OH on carbon 6 that's coming in and attacking, and the stereochemistry of the glycoside bond at the anomeric carbon is alpha. Okay, so it's down, it's that axial position. Okay, so that's why this is called an alpha-1,6 branch point, in addition to, of course, all of the alpha-1,4 linkages in each chain. And amylopectin is characterized by branching occurring at intervals of about 20 to 25 glucose units. Okay, so it's not branched, it's not highly branched, but it still does form these branch structure. Glycogen, on the other hand, is the carbohydrate reserve for animals, so humans, of course, being included. So if you have extra sugar in your body, more than your brain needs for fuel, what happens is that sugar gets polymerized into glycogen units. And the structure of glycogen looks a lot like the structure of amylopectin in that it is a branched polysaccharide, except in the case of glycogen, the structure is even more highly branched, and you have branching occurring as often as every 6 glucose monomeric units. Okay, so then finally the last polysaccharide that we need to be concerned with is cellulose. Cellulose is the structural material in plants. Okay, so that's what makes plants be strong. It's what they're largely made of. And cellulose is not broken down for food. So we said amylose and amylopectin are broken down for food if the plant should need additional food. Those were linked by alpha-1,4 linkages. Cellulose contains D-glucopyranoside units linked in beta-1,4 fashion, which leads to very, very long unbranched chains. Okay, so these structures are essentially linear and they do not coil into helical structures the way the alpha-1,4 linkages do. And that should be a fairly obvious if you look at the chair conformation here. So in a beta linkage, the acetal oxygen occupies an equatorial site with respect to both of the 6-membered rings. Okay, so the anomeric C1 as well as the C4 on the next carbon. And if you just think about what this would look like in 3 dimensions, it's going to form straight lines and not actually, you don't have anything more of the molecule underneath, which is what leads to the curvature in the alpha-1,4 linkages. Okay, so whether you look at the Haworth projection on the left or the cyclohexane chair conformations on the right, you can see that the CH2OH coming off of C6 and the acetal coming off of C1 are both up. They are cis to each other, and that's what defines a beta linkage. So plants have enzymes that can break down amylose in order to release it as energy. We who eat plants also have enzymes that can break down amylose. Starch is very easily broken down in your body into sugar. Cellulose is not very easily broken down. Many of it ends up as roughage that passes through the digestive system. Okay, because we have the enzymes that we have, the amylase enzymes that can break down amylose, can differentiate between the alpha-1,4 linkages and the beta-1,4 linkages, which is the only difference between these 2 types of polysaccharides. Okay, so we've gone through our tour of the carbohydrates, starting with monosaccharides, dealing with their classifications and their stereochemistry, putting monosaccharides together with other things to form glycoside bonds, putting them together with another monosaccharide to form disaccharides, and finally taking this and taking it to the nth degree and forming polysaccharides. So now that we're done with carbohydrates, if we continue through the organic chemistry of the biologically relevant small molecules, the next stop on our tour is going to be the lipids.